All right, another AP Biology review, second part of Unit 6 about gene regulation. And then we'll do some biotechnology thrown in at the end there because it kind of just fits in this spot. Starting the idea of this video with the idea that cells in your body are different because they have different jobs. But what actually makes a liver cell different from an eye cell, especially when you think about the idea that every cell has the same genes, has the same DNA. It all came from the same original zygote, the first cell. And you've grown through mitosis, and mitosis makes exact copies. But we don't want our liver cells to be exact copies of our eye cells. We need our liver cells to produce something called albumin, which helps to regulate all kinds of different things. Tonicity in your blood is one of those. And we want our eye cells to create something called crystalline, which is this protein that ends up being kind of see-through, and it's part of what makes the lens of our eyes, right? And so what we want to do is turn on the genes for albumin in our liver and turn off the genes for crystalline in our liver. But then in our eyes, we want to turn on the crystalline genes and turn off the albumin genes, right? And so this is called gene expression. But before we get into that example, this part starts with prokaryotic gene regulation. This is a simpler version of regulating our genes that you might remember about an operon system. The basic idea is that, especially on a single cell level, the cell really has to control its energy expenditure. You don't want to waste energy making things that you don't need. And so we can control the proteins that are in our cells based on are they being transcribed or not. If they're being transcribed, then you're going to get those proteins. If they're not transcribed, then you won't get those proteins. This is the primary level for eukaryotes as well, where we will regulate our genes. The operon system involves the genes of interest being connected to a promoter, which we already knew about. But now there's this thing in the middle called an operator that separates the promoter from the genes that we care about. And then there's this other gene that has its own promoter in regulation called a regulatory gene. The regulatory gene is going to produce a protein that then either fits or doesn't fit onto the operator, if you remember. And so the tryptophan operon is a normally on gene because tryptophan is required for the cell at all times, right? So most of the time we want RNA polymerase to be able to fit onto the promoter and then transcribe the genes, and that transcript will go off to find a ribosome to make the enzymes that then make tryptophan. So as a consequence of this gene being on, you get tryptophan to pile up. But eventually you might have enough tryptophan that you don't need it anymore. And at that point, making more and more and more might be a waste of energy or a waste of materials. And so as the levels of tryptophan go up, that leads to them having a higher chance of connecting to the regulatory protein. Right? And so remember, that was produced at some other time independently of what's going on here. And so the regulatory proteins is floating around. And as we now know, when a ligand connects into a receptor, it's going to make a conformational change. And so suddenly the protein is a different shape, and that shape allows it to connect to the operator. And so now, when the RNA polymerase is trying to do its thing, it can't physically get past the operator because we have blocked the operator with that protein. Oh, it stopped eventually. But while that's being stopped and the RNA polymerase is futilely trying to transcribe the genes, your body or the cell, it doesn't have to be a body, it could be a cell, is using the tryptophan to make other proteins. So the levels of tryptophan go down. This changes the equilibrium. Eventually, that leads to the tryptophan that was connected to the regulatory protein to fall off. Of course, when it falls off, the shape changes back, and the new shape, or the original shape, is not capable of connecting to the DNA operator. And now the RNA polymerase is free to go off and make more transcribe the genes to make more tryptophan, and then we're back where we started, right? Just to remind you that this is something called a negative feedback loop, where the production of tryptophan eventually turned itself off until we needed it again, where it basically turns itself back on. So that is called a repressible operon, right? Repress means stop. If we're going to stop this, that means it must have started to be on. So that was a normally on gene that is then repressed through the repressible operon. Now, sometimes you might have a protein that you only want to make in certain situations. This is the opposite of tryptophan, where you don't need it most of the time, but sometimes it might be a good strategy to have it. And so lactose is a sugar that lots of bacteria and other cells can digest. But you might not always find this as a diet. Like you're a bacteria, you might not be exposed to milk. So you don't want to sit there and make all these lactose digesting enzymes when you don't have any lactose around. So this gene is normally off. And now we know that off means blocked. So the repressor protein starts in a conformation that allows it to block the RNA polymerase. And if you can't get the RNA polymerase to access the genes, then you don't get any genes. You don't get any transcript, which leads to no proteins. But then lactose might become available. And once there's enough lactose available, the statistics, the probabilities that one of those molecules will find its way into the repressor protein, changing its shape like we now know, and the new shape can't bind to the operator. Polymerase is free to go and do a transcribe, and now eventually we get genes that digest lactose. But then here again is our negative feedback loop, because now we're going to start to eat the lactose, which makes its own concentration go down, which leads to a conformational change in the repressor protein and a rebinding onto the operator. And now we stop making those enzymes because the lactose is gone. And now we're back to that original situation where making them is not a good use of our energy. Right? So this is called an inducible operon. We can induce it. We can start it to happen because usually it is off, right? And so it's pretty simple in prokaryotes. We're just going to block or unblock the area of the DNA, separate the promoter from the genes, and that's the whole switch mechanism. These are the types of things that you're probably going to have on your notes, so you don't really need to memorize all these words anymore, but those are the major players. Now, eukaryotes still basically use the same idea. We want to initiate our transcription as our main way of controlling our DNA or our gene expression. But we do have a lot more opportunities to do this based on our situations. And of course, the ability to adjust to more diverse situations in different and more appropriate ways is a great strategy for evolution, right? So of course, eukaryotes have taken this path where things get more complicated, but this leads to a more complicated life. Rather than just being a simple bacteria that just turns things on and off. Now, one of the things that for a multicellular organism, especially that has huge DNA, huge chromosomes and tons of DNA, is that sometimes there might be huge sections of your DNA that you don't want to use, and we're never going to turn them on. So let's permanently shut these things off. Right. And so one thing that's interesting is that our DNA is so complicated that we can't just let it be super loose, like a completely loose spaghetti noodle. We got to wrap it around these proteins called histones. And the histones can be tightened or loosened. So in this picture right here, the DNA is loosely wrapped around the histones. And so there are sections of the DNA that are exposed, and we can use those pieces of the DNA to transcribe them. But then over here, we could pack them super tightly. We could make the histones coil very, very tightly. This DNA is not going to get transcribed. You can't physically fit anything in there, like an enzyme, to get in there to read the DNA. So this is a way to completely just shut off genes that you will never use. Obviously, bacteria is not going to have a whole bunch of genes that it's just never going to use. So bacteria and prokaryotes don't have this option, but we do. You shouldn't have to know this, but we just add molecules. We add acetyl groups. That's what those little green dots mean. The green dots will keep the acetyl groups, keep the histones from getting close to each other. You remove the acetyl groups, and now the green dots aren't there, and now they can go back and they can clump together again. There's another thing called DNA methylation, which is kind of like the histones, but this is going to put a chemical tag onto the Cs, the cysteines of the DNA. And these are going to function as like roadblocks that are going to keep the DNA polymerase from really binding and being able to read that DNA. One way that people kind of analogize this is by saying it's been redacted, right? Like, so you could have your DNA sequence that starts, and there's this whole section right here that you're using, and you use it all the time. But then maybe I just want to shut off just this one section of the DNA, right? And so we put the methyl group there, and now the RNA polymerase can't read that section. And then we can permanently turn off smaller sections of our DNA rather than just the big chunks of the histones, right? So accessing the DNA is the theme of those. Then we have the part where we actually are going to control transcription. And so here's our version of the operon system. We have control elements, some that are far and some that are close. These are kind of like the operator in the operons. Things are going to bind to those places sometimes, and if they do, then something happens, and if they don't bind, then it won't happen. One of the interesting things about our gene or our DNA here is that these far away regions are going to make the DNA flip into this section, right? So the reason why these are really far away is that we don't want any like what they call crosstalk. We don't want molecules to accidentally bind here. And so we keep the enhancers, the control elements, far away from the gene. But then that means that when you actually do get the correct molecules to bind to that spot, then you get this weird flipping action and you get this U shape. When everything is there, when all the different varieties of molecules are there at the same time in the right combination, only then will you turn on the gene, right? So this is way different than just like add lactose and it goes in there and it binds to something and turns it on. Now we need to add a number of molecules, and they have to be in the right concentrations compared to some of these other molecules that are all being regulated by way more complicated things than we can talk about. Here is the simplest way to imagine it, that you would have to imagine it, is that you have your enhancers over here. You have your regions that of the DNA that could bind the molecules, and they have like a code. You have the yellow, gray, and red. And if you get the yellow, gray, red molecule, then this part will get turned on. But of course, this will not get turned on because it's got a different set of enhancers, the green, gray, and pink, right? And so the availability of these transcription factors that you might remember come from outside of the cell, or sometimes they're generated inside of the cell. These are all controlled by all these different regulatory mechanisms. When the right situation is encountered, then the gene is on. When it isn't, it is off. Now, just one last thing to remind back to the last section, that in a real cell, you would probably never be choosing between these, right? I can't think of a cell that sometimes would want to activate albumin, but sometimes turn it off, and then sometimes turn on crystalline and sometimes turn it off. If this was an eye cell, this part would probably be methylated or completely wrapped up in the histone, whereas the opposite would be the liver cell, that this part would be methylated or the histones completely wrapped up tightly, right? But this is the main version. And so if you have questions about this, make sure that you get back to me. There are a couple other options that eukaryotes have that prokaryotes don't have because, again, they don't have the opportunity because they just use their messenger RNAs immediately, right? But we can control how long the messenger RNA lasts. Sometimes we want it to last a long time to get a lot of protein, or sometimes we want it to be shorter and not last very long so that we only get a little bit of protein. You might remember that is controlled by things like the poly-A tails. We also could use RNA, a fourth type of RNA called microRNA, to control the use of our messenger RNAs, right? These small RNAs, which you don't really have to explain where they come from anymore. This part about the dicer, if they wanted to do that part, they would tell you all about it, I'm sure. But the idea is that this little complex here that is has like an RNA homing sequence that will bind specifically to a messenger RNA. And this would then allow you to break down the messenger RNA or form a physical block to stop the ribosome. Either way, the presence of these microRNAs are going to shut down your gene expression, right? So you would use this in a situation where you've made messenger RNA, and for a minute there, that's what was the right idea, but suddenly it's like, oh no, we got to shut that down real fast. Then maybe these microRNAs come in and control that part. Sometimes you might go through the whole thing though, right? And you've already made a protein. So sometimes you want to take this protein that you just made and you want to break it into a bunch of million little pieces, right? So in the end, the final strategy to shut off your gene expression is to shut down the product that you already made, to destroy whatever it is that you did. One example that we have recently talked about would be the cyclins, right? Like when you want the cyclins to go away, you got to slowly break them down so that the cell doesn't stay in that stage of the cell cycle, right? So in the end, you have 5 kind of strategies that eukaryotes use. This is the one that's the most like prokaryotes, which is the one where you should make sure you understand it the most. The light in my camera is doing weird things, I just noticed, so sorry about that. Maybe that was freaking you out. Freaked me out a little bit. So here's where a terrible segue into the last little bit about biotechnology. We did 2 biotech labs. The first one about bacterial transformation. This relies on the idea that bacteria have separate pieces of DNA in their cells called plasmids, smaller little circles of DNA that actually get passed between bacteria. This is like bacterial sex. It's called conjugation. It's also something called horizontal gene transfer, meaning that it's not going up and down the generations like you would think of during regular reproduction, right? This would be a bacteria just like passing some information to another bacteria that is of the same generation as it is, right? And so that's the horizontal part. But what we figured out is that we could use these as vectors, that if bacteria are already passing these things around, let's get them to pass things that we want them to pass around. So that involves something called a restriction enzyme, an enzyme that cuts DNA at different places. There are all kinds of different places that you could cut the DNA if you have certain enzymes, right? They are all specific. So if you sequence your plasmid or whatever organism you want to cut up their DNA, you can look for the correct address to select the correct restriction enzyme, and then you can start to cut your DNA and paste it back together, right? So let's say you had these sequences, you would put the restriction enzyme and it would cut in a particular manner, and now you have separated your DNA into 2 pieces, right? And so let's say you have this section of human DNA right here, and you have the insulin gene, and you happen to know that there's a site to cut right there and a site to cut right there. You cut it, right? And now you've just made a fragment that includes your gene. Now your plasmid also should have cut sites that are similar, and so you cut that one, and now you have cut sites that match. And so if these things match, you can get them to stick together and use that stuff that we learned about already called DNA ligase, and it'll connect it together. And now you have made this thing called a recombinant DNA. It's been recombined between 2 different species. And so now the plasmid's ready for transformation. Don't worry about that, right? Now we can add other genes in there. Like antibiotic resistance genes is what we did. And then when you put them on the Petri dish, and the ones that live are the ones that have the plasmid, and the ones that die are the ones without the plasmid, because the plasmid has the antibiotic resistance. The plasmid has the thing that lets them live, right? And so this way we can figure out which bacteria took in the plasmid and then use those to do stuff like make whatever we want to make. Maybe we want to make a green fluorescent protein, which we also try to do. And now you can tell which ones got the plasmid, because not only did they not die, they should glow, right? And so this is just another way to use it. You can put whatever you want in there. We now put like insulin genes in there, like I mentioned already. Theoretically, you could get bacteria to start producing any protein you wanted them to make, because all you need to do is take the DNA and put it into the plasmid. And thanks to the nature of DNA, that all living things have the same genetic code, the bacteria don't know the difference, and they just, their enzymes just do what they always do. And there you go. this has jumped too fast. But we also did this other thing called gel electrophoresis, where we were separating DNA fragments. This is the second biotech lab that we did. It's based on the idea that since everyone has different DNA, there should be different places to cut DNA. So the idea here is that if I take these two pieces of DNA that look the same on the surface, I cut them, and now there's different fragments, and the fragments are a pattern, and you can compare the patterns to see what identify an individual, right? So you take this stuff called agarose gel, and you it's like Jello, and you put it into a mold, and then you add your electrical field to the mold, and you add your sample. And so the DNA is negatively charged, and it's always going to go towards the positive pole. Smaller fragments will move through the quote unquote Jello faster than large fragments, just because of the physical properties of being large, trying to squeeze through the little pores in this gel. And so this is a way to naturally separate fragments by size. And again, that's what gives you that pattern. So you put your samples in the wells, and then you add the electricity, and you wait for them to move. And they won't look different. This one is going to be the same visual as this band, but the molecules that made this band are much smaller than the molecules that make that band, right? And so again, this is separating by size, and now you can look at things and say, oh, this one, it has a pattern that looks like that with those 4 sizes. And that's very similar to this one, although there's a carryover there, and not so much there, not so much there, right? So like you can compare the patterns, right? And this is all based off of just the physical properties of DNA being negatively charged, and the fact that smaller things will move through this faster. And so again, so here's maybe an idea where you would use it, where you could identify somebody who has a sickle cell mutation, because the mutation removed a cut site right there, right? There's a cut site here that isn't in the mutated version. And so somebody who has the mutant sickle cell gene, they're going to have a different pattern than the person who doesn't have the mutation, right? So this is, so maybe you wanted to just verify that somebody had a mutation. You could get their DNA, run it through the restriction enzyme, put it through the gel, and then you have now identified at least the pattern that it makes, and then go from there, right? And so this is one way that we actually confirm the results when we are doing the like the bacterial transformation. You do all this work, but you can't see what's happening, right? You just have to trust that the liquids are doing what you think they're doing. And so once you finally get it and you say, oh man, it glowed. Well, let me make sure that it's really because of what I did. And so you could go in there and you could cut the DNA, because now you know the way it should cut, and then you run it on the gel and you compare it to like the original one that you did before you tried to transform it, and see if they match or don't match, right? So in any case, this is the end of the unit, and I think this is the last unit. So this is the last video of me creepily getting weird lights on my nose that I can't notice. But sorry about that. But good luck, and let me know if you need anything.